So again, my talk is still on the biological tissues, and there indeed, and there are biomarkers found in biological tissues like prostate cancers, and the citrate is a clinical marker, and being used to clinically to differentiate the people has prostate cancer with whether it has prostate cancer. And also in a number of cancer cases, like the colon and the prostate colon ratios, and also their concentrations, they are clearly biomarkers of cancers. So that's in tissues. So I will focus on tissue, the profiling. So this one I already talked about. So this is magical spinning. So we do not do tissue extraction. So we put integrate biological tissue inside the spinning MR rotor and do the MR spectral. So that's what you may get. So this one is very has some similarity with whatever you can get from doing tissue extractions. Tissue extraction can take like hours or more very easily. So for this kind of work with intact biological tissues, it may take only about 15 minutes, 10 to 15 minutes to get a spectral. Then you can be utilized to say the signature and peaks to say whether and the patient or the studied case is diseased or not diseased. For this type of experiment, they are very powerful. The only disadvantage for this one, not a disadvantage, just something you have to spin the samples. 1 second, you rotate the sample several thousand times inside a strong magnetic field. Since fast magical spinning, you cannot use a rotor that's constructed of metal, so you have to use ceramics or plastics. So to produce 100% leakage-free samples for this traditional high-resolution magical spinning MR is not that easy, because commercial companies they provide rotors. You can be used for a few times, maybe seals the fluid inside the rotor or the tissue inside rotor pretty well, and after time, and it becomes very difficult to seal the samples. So the second difficulty is because you rotate per second several thousand times, the large centrifuge force associated with the sample spinning will destroy the tissue integrity and sometimes even lyses the cell. Also, it's difficult to spin pretty large sample volume, like 300 milligram or more, with the high-resolution MR, because the more sample you put the rotor, if you want to spin higher, the more difficult it will inject the plugs, everything, and very difficult. With small sample volume, also very challenging. It's difficult to put the sample in. We have methods that can make it work, but so it's difficult to put the sample inside the capillary tube and seal it with a short period of time. So our research objectives is we wanted to provide high spectral resolution and on integrated biological tissues, just like the traditional fast magical spinning, and is capable of non-minimum destructive detection, can work with a range sample size from very small to very big, 1 millisecond using a single probe. How we do it? So we combine a technique called slow magical spinning. So that means we rotate the sample very slowly. And we also use a switchable inductive coupled static micro MR coil resonator. So that's the slow manual spinning methods. Basically, they are train of pulses, and we synchronize the sequence to certain road periods and generate a two-dimensional spectrum. The problem is if you spin very slow and you do not have a spectrum, you generate a train of sideband. So with this sequence, we can spread in the two-dimensional spectroscopy and select the high-resolution spectrum. So this is an example on tissue samples. This is brain tissues with a spinning rate of only 43 hertz. So that's 1 second you rotate the tissue 43 times. You do not lysis a cell in this case. And for muscles, for livers, for those tissues, and the tissue integrity will be kept. So this one is our latest development. So we how to do small sample and big sample with a single probe head. So this is basically this is a small coil resonant around the sample tube for small coil detection and inside the large coil. And when you remove the small coil, and you can put a big sample tube here and work with large sample size of tissues. And this is how we engineering the microcoil resonator and put a small coil inside, like the coil has 1 millimeter ID, and with a coil wire of 0.1 millimeter, it's not easy. So you have to have a very special way to put the coil inside and build a resonator and to do the microcoil resonance. So that's a feature of the probe. Basically, a microcoil resonator is inductively coupled to an outside coil, just like a transformer. It works. And in this way, and a very small sample inside, and we have very high sensitivity because the filling factor is high. So we did theoretical simulations. So that's without microcoil, that's the field strength inside the coil, and with microcoil, and looks like the flux is just concentrated, kind of focused into the inner one. That's why we have increased the sensitivity about 20 folds. So that's we can do different combinations and find out the optimize the geometry. And with a different magnetic field, if the inner coil resonant like 300 megahertz, inner coil resonant about 305 megahertz generate maximum enhancement. 400 megahertz, it's about 405 also, and 500 megahertz becomes changed. You really need to shift the resonance of inner coil to 520, 530 to get the maximum enhancement. So in practice, we build the coil, we test it, and indeed we get enhancement about 22-fold. With such an enhancement, we can do very small sample volume, like 200 nanometer. That's about 2.2 milligram, and with a few minutes. So that's one kind of biological tissues. That's the muscles from mouse. We get a bunch of metabolite here. So using this little time, we can also put a big sample volume. So that's the several mouse brain. We pull it together, put it in the rotor. If we just do static, you don't have spectral resolution. If we rotate it 83 hertz, we get this kind of resolution. So this is all about spectral resolution enhancement. Then this technique can also work with various small amount of the biofluid. That's a urine sample. So when you go to the traditional MR metabolomics using this kind of sample tube with very small sample volume, and you have a bunch of features, but you have lots of non-broadening due to the sample tube. When we spin very slowly with the slow manual spinning methods, we get a spectral resolution similar to whatever you can get with the traditional high-resolution MR with about 100 microliter of the urine sample, so very similar. So we can also work with naive small biological objects. In this case, as in the situation, this is a garden bug, pill bug. So we can put either rotor, spin it to 82 Hertz. So that's 1 second, rotate it 82 times. We generate the metabolite spectral here. And the top is we spin 4 kilohertz with force manual spinning, something very similar on this case. Of course, in this case, this one is not destroyed, but with this kind of spinning rate, after the experiment, when we get the bug out, it's already widely dead. So this is about we report methods. So we can work on very small sample volume, about 200 nanoliters to 1 cubic centimeter, using a single probe. And the nanoliter features may have the potential to follow the metabolic changes through continued investigation on a single small laboratory animal over long periods of time, like using blood or tissue, like a tissue biopsy sample. We can use needle biopsy samples. The milliliter features would allow minimum destructive study of integral biological object with size as large as 1 cubic centimeter. So this one has the potential and to go into clinical settings, because when doctor cut off something, and you can quickly put it inside the probe, and within in about a few to 10 minutes, you get the spectrum. Then you get some signatures, so you can decide and some what's the disease state of the exercise tissue. So, so these people involved in this study. I do have a couple of biologists as collaborators. So, so this is the grant from NIH to support this type of work. And so this is conclusion of my presentation. And thank you for your attention. And I will be glad to answer questions. Thank you.